All right, welcome back. We're in chapter 17 on protein synthesis, more or less. Information in DNA is stored in the sequence of nitrogenous bases like the A, T, C's, and G's. Um, that, that code, that information is passed down from generation to generation. That's why we call DNA the heritable molecule of heredity. Um, so, like ourselves, we express those genes by producing proteins, and then the proteins give us traits in different ways, shapes, and forms. That's in a two-part process of transcription and translation. This is mainly going to be about transcription. One of these things is not like the other. Here we have albinism in some sort of deer, and that's going to be because there's some sort of mutation in the DNA of the deer on the left that's not allowing it to produce much, if any, pigments. So how do we figure this out? Well, you see, what it was Beadle and Tatum took mold and they zapped it with X-rays, and they came up with what's called a one gene, one enzyme hypothesis. So let's take a look. Here so they have you have kind of two types. So you got your regular mold, and that's what's called the wild type, right? So what we call normal. And then they've got like mold that they hit with X-rays, and when they try to grow both of them on what's called minimal media, and that's like a medium that doesn't really have a lot of nutrients in it. It might have some bare bones nutrients, but not enough where if you're missing certain biochemical pathways, you're not going to be able to survive. So what they found was they've got these mutants, if you will, these mold that were kind of exposed to X-rays, and what they found is that some of them, if you kind of grew it on minimal medium, but then you supplemented it with ornithine, which is kind of like an amino acid. If you supplemented it with ornithine, they were fine; they could grow. Not only could they grow like an ornithine, they could grow if you had citrulline and arginine. All right, some of them. And by the way, so this is a biochemical pathway. If you take ornithine, you can convert it into citrulline, and then you can convert that into arginine. So the idea is that this class 1 mutant, the enzyme that's missing is the one that takes kind of our precursor and makes like ornithine. All right. So the X-rays like deleted that gene or made it inactive, made it not working. Okay. So the mold can't grow in this minimal medium, but if you just kind of move it down that biochemical pathway, you add ornithine, then it's fine. All right. So then the same with the class 2 mutants, except if you supplement with ornithine, they still can't grow, but when you throw in citrulline, it can, and that's whatever enzyme is here. Wait, I can use my pointer. Whatever enzyme's here that converts ornithine to citrulline, that's knocked out. Much like this enzyme here, and the first class 1 is knocked out. Class 3 mutants, whatever's taking citrulline and converting into arginine is missing. All right, so this is how they kind of realized. All right, well, for each gene we have, they're making a certain enzyme that either can do biochemical pathways or whatever else. So originally it was the one gene, one enzyme hypothesis. Later on, it turns into the one gene, one protein hypothesis. Here's kind of an overview of what I was saying. Whoops, not there. So Gene A is responsible for enzyme A, which takes our precursor and turns into ornithine. Gene B is responsible for enzyme B, which takes ornithine and turns into citrulline. And then gene C is responsible for enzyme C that takes citrulline to arginine. Okay, and then our class 1 mutants are missing enzyme A, or I'm sorry, gene A, or functioning gene A. Our class 2 mutants are missing a functioning gene B, and hence a functioning enzyme B, and then same with gene C. One gene, one enzyme, or one gene, one protein. And that this is all I was saying. The revision is that not all proteins are enzymes. Later on, they figured out it's one gene, one protein, and so on and so forth, or one gene, one polypeptide. It's all the same. So, how do we go from DNA to protein? Well, first we have this process called transcription, and what we're doing is we're taking DNA information, we're turning it into RNA information. Okay. That DNA information ends up producing something called messenger RNA, or mRNA for short. After transcription is done, then we do translation, where we're taking that RNA information and turning it into a protein. And this is all happening in the ribosome, whether those ribosomes are in the cytoplasm or in the rough endoplasmic reticulum, depending if you're a eukaryotic or prokaryotic cell. We're going to mostly do the prokaryotic version of this, but keep in mind that they're slightly different. Mechanics are basically the same. All right. So in the prokaryotic cell, basically we have transcription off the DNA. We make mRNA, and then we do the translation. And this is all in the cytoplasm. In the eukaryotic cell, it's kind of cool. You make you do the transcription. It's in the nucleus. Then it leaves the it gets processed. There's these things called introns and exons. They do a lot. They do like splicing and whatever else, and then they add a portion to the front and a portion to the end, so that the head and the tail. Then it leaves the nucleus to go to the ribosomes, whether they're in cytoplasm or in the rough endoplasmic reticulum. So all that happens in the nucleus, the transcription, the processing, and then the translation occurs in the cytoplasm. So our genetic code is in what we call like triplets, and the genetic code's all found in the or it's all read in the messenger RNA. You have 20 amino acids, 4 nucleotides, right? A, T, C, and G, or in RNA it's A, U, C, and G. Okay, so you got 20 amino acids, and we're looking at them in triplet code. Well, if we have triplets, that gives us 2 times 2. No, sorry, like 2 to the 4th times 2 to the 4th times 2 to the 4th. Basically, we have approximately like 64, I want to say, possibilities for only 20 amino acids. So here's our actual chart. We call this a codon chart. And if you look, so here we have like UUU and UUC, and they both code for phenylalanine. UUA and UUG both code for leucine. If you start with a U and a C, you code for serine. All right, so we have a lot of like repetitiveness or redundancy. Some other features on here is you see we have a start codon, and that start codon is a start, and it also codes for something called methionine. We have 3 stops: UAA, UAG, and UGA. The stop codon tells the cell to stop making the protein, much like the start codon tells the cell to make a protein. All right, so here you can see this kind of an overview. We take our DNA, we open it up, we do transcription, all right, and then we do translation. All right, so transcription and the mRNA, we have the codons. So whatever is happening, if we have an A, we make a U, or we lay down the complementary base, which is U. If I have a C, lay down a complementary base of G, C, G, and so on. A with U, U, We put a G, G, C, and so on. All right, and then if you read the codon, so for instance, UGG, if I go here, so U was our first base, right? So U, and then I scan over to G. So all four of these start with UG. UGU, UGC, UGA, and UGG. So UGG codes for tryptophan, all right, which we have right here. UUU, so I have my U, U, and then U, codes for phenylalanine, right? Here's phenylalanine down here. GGC, G. See, this is the second base is G, right? So GG, C, or glycine. Whoops. And so on. And until we get to a stop codon, and then we translation would stop. So that's kind of how you read the codon chart, which is an important skill to have. So, you know, and this codon chart is universal. It's not just for like people, and the bacteria have their own. It's like our codon chart is the same as bacteria. It's the same as a slime mold, same as a fungus, same as an algae, and so on. And because of that, we do these really bizarre experiments where we take, like, say, the gene for lightning bugs or fireflies that does the glowing, and you can implant it. You can put it into a tobacco plant, and you can make tobacco plants glow in the dark because you know, we have the same code. You know, if we splice in that lightning bug gene into the tobacco plant, the machinery within the plant can still take that gene and express it. All right, and then here, I can never really understand this one, but yeah, they took jellyfish gene and put it in the pig, and it gives the pig, like, I don't know, it gives them like different pigmentation, I guess. But yeah, these pigs look kind of weird, and I really have no clue why somebody would do that, but they did. Maybe they just because they could. I don't know. I mean, I kind of get this. I mean, that's kind of fun. This would be really cool, right? You could plant them in your yard, like along like a sidewalk, and then it would light up your sidewalk. Just saying. Yeah, you got me. But anyway, so transcription. The first part of protein synthesis. Let's take a look at it. Our enzyme is RNA polymerase, okay? And RNA polymerase actually kind of acts like DNA helicase. Actually, it acts like DNA helicase, the primer / primase, and then DNA polymerase. Like all from like DNA synthesis, it acts like all of them. Basically, it just comes along. It finds something called a promoter. All right, so the promoter is kind of the area where the RNA polymerase is going to bind to. Yeah, so you can see from this picture here, it's like a starting point. All right, so it finds the promoter, and our gene is going to be down what we call downstream from the promoter. All right, so it opens up the DNA, and it starts making the messenger RNA. Your text, and I guess nowadays they call this initiation, elongation, and termination. Thanks, Tom. And I can tell you, I remember when there was a time when these phases didn't really exist. So I generally don't refer to them except in protein translation. There's a clear initiation, elongation, and termination. So in this one, initiation is where, like, you know, you're bound to the promoter, and then the RNA polymerase starts to open up the DNA and starts to make the messenger RNA, and then elongation is where the messenger RNA is being made, and then termination is when the RNA polymerase falls off. So this last slide here, you can kind of see part of what would be elongation. So, you know, we've got the DNA opened up where we're fitting. It's like we've got part of our messenger RNA hanging off here, and it's like, okay, we get to an A and we're gonna fit. I'm sorry, we get to a T and we're gonna fit an A with it. Okay, we got an A, we're gonna fit a U to it. Oh, we've got another A, we're gonna fit a U to it. Battery's running low. We get to that C, we're gonna add a G, and so on. Okay, so and once you get to the end, and then the RNA polymerase falls off, you're done with transcription.